Hey guys, it's Lauren, Natasha.
Tim.
Welcome to BS, Biological Sciences. This will be our first video and our last video. Today we will be talking about light-dependent reactions.
The whole process begins when photons, or energy units, of sunlight strike a leaf, activating chlorophyll and exciting electrons. The activated chlorophyll molecule then passes these excited electrons down to a series of electron carriers. The whole point of the light reaction is to produce energy in form of ATP and electron carriers, specifically NADPH. So as you see here, the light enters the chloroplast and excites electrons in photosystem 2, which goes into the electron transport chain and then to photosystem 1. This creates ATP and NADPH. These two molecules are used in the Calvin cycle along with carbon dioxide to produce sugar.
Many light-absorbing pigments participate in photosynthesis. The most important are chlorophyll A, chlorophyll B, and carotenoids, which are clustered in the thylakoid membrane into units called antenna complexes. All of these pigments within a unit are able to gather light but are not able to excite the electrons. One special molecule located at the reaction center is capable of transforming light energy into chemical energy. Basically, the antenna pigments gather light and bounce the energy to the reaction center. As you can see here, the light enters the thylakoid and hits the antenna pigment molecules, which then transfer the energy by bouncing it from one antenna pigment molecule to the next antenna pigment molecule, and then they bounce it all the way to the reaction center chlorophyll, which then sends it to the primary electron acceptor. There are two types of reaction centers, photosystem 1 and photosystem 2. The principal difference is that each reaction center has a different type of chlorophyll. Photosystem 2 has a reaction center called P680, and this has the maximum absorption at the wavelength of 680 nanometers. Photosystem 2 has a reaction center called P700, and it best absorbs light at the wavelength of 700 nanometers. When light energy is used to make ATP, it is called photophosphorylation. So as you can see in this picture, in photosystem 2, there's a high energy of electrons, and then once you head to photosystem 1, you have a decrease in energy because the proton gradient is formed for ATP synthesis. And then once you reach photosystem 1, there's an increased energy of electrons again to then help produce the NADP reductase.
The non-cyclic method of photophosphorylation produces ATP using both photosystem 1 and photosystem 2. When a leaf captures sunlight, the energy is sent to the P680, the reaction center for photosystem 2. The activated electrons are then trapped by P680 and passed to a molecule called the primary acceptor. They are then passed down to carriers in the electron transport chain, or ETC for short, and enter photosystem 1. When the reaction center P680 absorbs light, it also splits water into oxygen, hydrogen ions, and electrons. That process is called photolysis. Hydrogen ions accumulate inside the thylakoids and a proton gradient is established. Essentially, in this diagram, it shows that the first step is photosystem 2, so you need a primary acceptor for that, and an output of this photosystem 2 is ATP, and this ATP is used for chemiosmosis. Then you need photosystem 1 and another primary acceptor to then eventually reduce NADP plus to NADPH.
The cyclic method uses a much simpler path to generate ATP. The electrons in photosystem 1 are excited and leave the reaction center, P700. Then they are passed from the carrier to carrier in the electron transport chain and eventually return to P700. This cycle only produces one ATP. It is not a very efficient method for production of energy. This pathway also does not produce NADPH, and plants use this only in a method when there is not enough NADP molecules to accept electrons. So in this picture, we see the cyclic method, which is basically a simplified non-cyclic method. The electrons are excited and go to the primary electron acceptor, which is then converted into ATP, but then the electrons go back to photosystem 1 instead of traveling on to photosystem 2.
In review of non-cyclic phosphorylation, P680 in photosystem 2 captures light and passes excited electrons down an electron transport chain to produce ATP. P700 in photosystem 1 captures light and passes excited electrons down an electron transport chain to produce NADPH. A molecule of water is split by sunlight, releasing electrons, hydrogen, and free oxygen. In review of cyclic phosphorylation, P700 in photosystem 1 captures light and passes excited electrons down an electron transport chain to produce ATP. NADPH is not produced and water is not split by the sunlight.
So overall, we learned about light-dependent reactions. We talked about how the whole process begins with photons using chlorophyll A and B in the reaction center. Photosystem 1 and photosystem 2 are both reaction centers. You have two methods of the light-dependent reaction, non-cyclic photophosphorylation and then the cyclic method. Thank you, and we will see you next time. Oh wait, there will not be a next time.